this is important information, even though it's probably the most difficult and confusing part of setting up a program, but at least you'll have that as a background. I don't have an answer. I can't tell you this is how you're going to enumerate. I can tell you how I do and would recommend it, and some states do that too. But these are some of the issues regarding enumeration. And as you saw from those pictures, those photos, you got this massive colony of cells with tens of thousands of cells in a colony. How many colonies do you count for statistical significance or anything else? And there are some, I'll give you some guidelines, but you'll have to, you'll have to involve the people who understand, statistical and reports, and whether you have an epidemiologist in the group that helps with that kind of thing, would be important. But I can give you the basics, and then again, I want to give you the specific basics for the algae to identify today, cyanobacteria. So what I'm doing here is goals and problems for enumeration methods, tools, recommended approaches, and using those results for setting action levels. Whether it's a flow chart or whether it's how you get to a point where you say, okay, now we have to post this lake or put out some kind of an alert. And then one of the things that, I was told not to include water treatment methods, anything like that, because that's actually, I think, what you'll cover next year, perhaps. And then at the same time they said, but, cover it anyway or something. So there's a couple of summary slides in here from the international perspective of WHO and what works and what doesn't work. Okay, here's the problem. And this is an extreme problem, but it's one that I encountered, and if you have to make these decisions, maybe you do too. This is an RFP for enumeration from the city of Salem in Oregon just a year or two ago, last year I guess. And this is what they put into their RFP saying they wanted. Okay? They wanted the capability to analyze freshwater algae, phytoplankton and paraphytic, analyzing samples for heterocysts and aconites, and conducting standard analysis on preserved samples, and they're recommending Lugal's. That standard sample analysis includes, can you hear me? Okay. Identification to group and species level, density, cells per mil and percent, biovolume and cubic micrometers and mils percent, density in cells per mil for all toxic species, total density and total biovolume, and a trophic level index. Biovolume estimates encountered within a minimum of 100 natural units. We haven't discussed natural units yet, but as an alternative to counting cells, if you count a colony, that's a natural unit. Electronic data in Microsoft, compilation of results, spreadsheets, sample dates, all this information, and then they want a 4 to 12 week standard turnaround time, a 1 to 2 week fast turnaround time, and a 12 to 48 hour rush turnaround time, and all this for $1.95 or something like that. So this is an extreme, but these are the things you have to decide when you go out and say, how am I going to monitor? What am I going to monitor? And obviously, this gets significantly reduced. But this is an example. I declined. I didn't respond to this RFP. I have another life to live. But somebody did, and I'm not sure what they ever accepted. But to fulfill this budget would be hundreds of thousands of dollars. But some typical questions agencies that do need to basically answer, which I can help give you. In order to obtain a statistically sound cell count, what is the lowest number of cells one can count? Do I have to, if I have a sample and I count 1,000 cells, is that enough? 100 cells? 10 cells? Will I get a representative, statistically significant number? After the lowest number has been reached, is it correct to assume that this is when sedimentation will be formed? In other words, if I take a raw water sample and count it directly, and I end up with certain cell counts, this may drive me to say, okay, well now I have to sediment that sample, concentrate it, whether it's 100 mils or 500 mils or or whatever. So these are the kinds of things I can help you with. Some of the key references for that kind of information, we've already alluded to these. This is Chapter 12, which is in your lab manual. Chapter 2 is the AWWA manual, this one. The sample collection and standard methods, sampling methods and setting guidance in the state of Oregon. After their CDC study, they had all of this, and that's on the website too. So there are places to get started with. Australia, as I implied, has done a lot of this already. They have a digital book on phytoplankton methods manual for Australian freshwater. Those are all available online. So this is what we are coming down to then. How do we count these filaments and these colonies, and if they're there, individual cells, which I don't see any. Okay, here's a filamentous, Lingbia. This is Lingbia. Fairly easy and straightforward because they're long, very large filaments. These are large in the sense that they're probably 20 or 30 microns across, 5 or 10 microns in width, and it's fairly easy to count those cells. This is not as easy as it does get. And you would place these in a cell counter, and you would count whatever you see on your defined grids that you decide is statistically significant, and then you can go from there and get cells per mil. In the background, though, you'll see here's another filamentous cyanobacteria, much smaller. That's probably an oscillatoria, and so you'd have to count it as well. But how do we count this? These are globs of microcystis, and often this is what you end up seeing. And these individual globs will be comprised of lots of colonies. So how many of those do you count? This is Aphanizomenon flos-aquae, kind of from an underwater camera. And here's our dilemma. These are the colonies, the flakes, typical for this species, and each one of those flakes is comprised of filaments, and each filament is comprised of hundreds of cells. How much of that do we have to count? Looking a little closer then, these are those colonies of Aphanizomenon. So this would be one colony, that would be another colony, and at this level, you can barely even see the cells, and this is probably just 100 magnification. All right, not to scare you away, but this is what happens. Keeping in mind those colonies we just looked at, a single filament of AFA typically ranges from 3.9 to 4.4 in width. An average of 10 filaments was actually 4.3 micrometers. And it ranges in length from, say, 60 to 300. Average of 25 was actually 128. They're large and difficult to measure a number of filaments in any of the above measured flakes. Now we have to go a little bit lower and say 1,000 micrometers in a millimeter. Side by side, there would be about 230 filaments lying parallel with each other in one plane. But this is a three-dimensional colony. So now we have to say that in a flake that is 1.6 centimeters in length, which is 16,000 micrometers, there could be about 29,000 filaments lying in the one plane, single plane. And then there are multiple planes or layers. And the flake thickness could be about 1/10 of the width, which makes it 100 micrometers, and which could accommodate about 20 layers, depending on the individual width of those filaments. So that affords a colony size of maybe 500,000 filaments, and each filament several hundred cells. So you're not going to count that. I'm not going to count it. But that's where we're at. And of course that's one colony. And in a mill you're going to have few hundred colonies. Well, you're not going to. I guess that's the answer. But there are ways to kind of get enough out of it. So what do we use for enumeration? In terms of counting units, most people think of cells, but when you're dealing with something like this, it's becoming more and more common to start dealing with what's called natural units. So a natural unit would be the colony. So you count colonies, not worry about how many cells are there. Because again, keep in mind, what are we doing this for? We're doing it for management decisions. And if we're all talking apples and not apples and oranges, then we can make management decisions based upon something that's not so onerous as cells. The other possibility is to use aerial standard units, which I'll explain in just a minute. And then out of that, we have to ask questions about whether we're more interested in precision or accuracy. And then the counting method. Are we going to count the whole field under the microscope or the counting chamber? Or might we just look at passes, you know, up, down, cross, whatever, or actual strips? So these are three ways. And of course, or the whole sample. And there are chambers that you could place the whole sample into and count everything in there. And that might be appropriate if it's big enough and dilute enough and gets you the answer that you want. But probably what we're going to be coming down to is looking at passes and strips. Out of the standard phytoplankton counting information that's in the AWWA, this manual right here, this one, there's a little table, and they summarize this as follows. The enumeration method, total cell counts, natural unit counts. Counting unit for total cell count would be the cell. The natural unit would be the organism, or in other words, if it's a single cell, that is the natural unit. But if it's a colony, it's the colony that's the natural unit. The reporting units would be cells per mil and units per mil. Another way that's kind of in between that is called the aerial standard unit equals the area of 4 small squares, and if you're using a Whipple counter, I'll show you what that is in just a minute, or 4 1 millimeter squares on a Neubauer plate of 200x. Then the definition, that would be the aerial standard unit count, which in this case would be, if it's 4 1 millimeter squares, then we're dealing with 400 square micrometers. And that gives you units per mil again, because you're using the natural units. And this, we're starting to condense down from counting so much to getting something that's reportable in a standard way. Okay, concentrating samples. Concentrating, as I mentioned, once you get to a point where you've only got about 10 to 15 cells in that undiluted count, you start getting into statistically gray areas. And so you need to up the number you count from 10. 10 or 15 is not acceptable. So how do we concentrate? Well, you can centrifuge, but centrifugation often disrupts cells, and especially planktonic cells like anabenas and microcystis, if they have gas vacuoles, then they're floating. You can't centrifuge them without literally destroying them. So what we do then is filtration. And the standard way that's always been around for filtration is something called the Udall chambers. And at the bottom of those you have a natural cellulose filter which can be cleared for permanent mounting or counting. And often they'll use an inverted scope once they take that disc off and put it on. If you've got the space on a top right scope like this, if you got the space in here, you can do counts that way too. It depends on your magnification. But because of that distance to the objective, you often have to go to the inverted scope. While the Udall chamber looks like this is one that I bought. You can buy in various places. And some people make them for a living, and some places sell them like Ward's and Carolina Biological and so on. This one's on a platform and has levelers. and a little sight bulb for making it level, because if you fill all of those chambers, here's a chamber, a chamber, a chamber, and a chamber, and if you don't have it level, then you're going to have different settling rates. And then you put a chamber on top of various sizes. So the biggest one would be about, I think this is 100 mils. This is probably 50, and then maybe 20, and then 5. And these screws come off. You put the chamber in there, and then you could have a filter disc, and then you attach the vacuum, and then here each one of them is vacuumed. So you don't have to, you can do it two ways. You could settle, typically standard is settle overnight or 24 hours. And, but of course most people would put a slight vacuum on it and speed that process up and filter. So that's how you get a standard volume concentrated to be able then to work from that concentrated sample and do your counts and work back to something that says cells or natural units per milliliter or per liter, whatever you're reporting. This is out of a standard methods book that describes the Outremal. Typically use an inverted scope. A bit about samples. Here they say 10% Lugol's, and so on and so forth. But you can use this for everything, you know, zooplankton as well as phytoplankton. Okay, what about after you've done that filtration and you take that off and you put it, what kind of a counting chamber are we going to use? Well, it kind of depends on, you know, whether you're dealing with how dense the sample might be, whether you're dealing with a lot of particulates in the sample, whether you've got a lot of color in the sample, the size of the algae, and the type of microscope that you have to work with, and so on, and of course how much time you have to pay somebody to do this. But these are all, you know, they're all doable things. So here's a few examples of counting chambers. Something called the Sedgwick Rafter counting chamber. It's just a slide, and they've glued some plastic on here. This one has a grid so that you can keep track of where you're counting, because believe me, your eyes start to wander, of course. But here's one that's not gridded, and typically these are 1 mil, so, and this is the case where you're counting everything that's in a mil, usually. I don't use that. Phycotech, the company that does a lot of these enumerations and identifications, she actually has built smaller standard size counting chambers, and if you buy one of Anne St. Amon's chambers, she has it individually calibrated for volume. I've brought examples of all these chambers, by the way, so you can look at them. In this case, this little chamber might be calibrated, and you'll get a calibration that says this is 0.3256 milliliters or something like that. So you count that and then calculate back to cells or filaments or natural units per mil. What I have ended up using is a little different. Here's a reference to that Whipple ocular, which traditionally and classically has been used a lot by the limnologists and phycologists as well. It's called the Palmer-Maloney, and it's a 10th mil volume, and then the ocular that you use is the Whipple ocular in the microscope, which gives you the statistical significance, and we'll stick with that in just a minute. This is what I have used, and I don't get any objections to it, and I think it works well. It's a little more expensive than one of these other chambers, but it's out of the medical use. It's literally a hemocytometer for counting blood cells. And it's a thicker slide. It has wells on either side. This isn't, but I'll show you the real thing. It's raised, and there's a trough on either side of this. You place the sample here and here, so you get replicates. So you can get 2 replicates. And then these are gridded. And then you count a certain number of grids on either side of that. And let me show you what that looks like. Here's the Whipple ocular, and typically you count units in 20 to 30 random small fields or 2 perpendicular transects. And of course, it's probably easier to count transects. So you might count the transect here and the transect here, and that would give you your 20 to 30 random small fields. So that's what's typically done. So now we're down to counting these smaller chambers or grids, and we count all the cells in there. On the Neubauer ruling for cells, this is what you see on either side of that trench on the Neubauer plate. It's nicely gridded, and that makes it more expensive. These are about $70 or $80 a piece, but they're, use them over and over and over again. Each little small line is 0.05 millimeters, and each bigger square, like this, would be 1 millimeter. And then you choose within that either grids based here or grids based here or things in here. It depends on, let's say you had a lot of little algae and you needed much smaller grids. To keep track of things, you might count those grids. And this might be how you would go about it. Choose 4, and we take the average of what's in those 4. And just be consistent, maybe start in the top left and then do this pattern down through the grids. Or the same if you were counting these grids. Keeping in mind that these will be smaller, but then you simply plug that into your equation for your back calculation. So this is how I would approach it. I would do 4 grids. Typically I like to do these, because usually when I get sent samples, there's a lot in it. I don't need to get down to the small level. So the statistical considerations of this, you need a unit density of about greater than or equal 10 per field to get 95% confidence. And that's usually not hard to accomplish. In other words, we need 10 cells or 10 natural units in those grids, in the field grids that we choose. And then we are assured of having 95% confidence. For the level of precision, in other words, how many units do we have to count or cells, if we want a precision roughly between 10 and 50%, the lower the better. That's 400 units to get 10% precision and 100 units for 20% precision. And typically, it's acceptable to go 10 or 20% precision on your cell countings. This just is the little formula that would give you that precision percentage. So as long as we're roughly counting around 400 units, and we've got unit densities of 10, greater than 10 per field, we're going to be well within statistically good areas for countings. Yes?
Okay, sorry, I'm just getting a little confused because we were talking about calling a colony a unit, or no?
Either way.
And now you're saying, well, I guess what I'm thinking is that if you have a sample that doesn't have very much in it,
well, then you're probably counting cells.
Oh, I see.
But if you've got so many that you want to count natural units, then it would be greater than or equal to 10 natural units. As long as the number you're counting is 10 or greater, whether it's cells or filaments or colonies. Does that make sense?
Okay. I hope it'll work when we sit down and
Yeah.
look at it.
Yeah. If you count 10 of something per field, and you have predetermined this is how I'm going to do my monitoring, whether it's cells or natural units, then you're okay in terms of statistics. Okay? Okay. Clear? So, summarizing that, this is what I came out of the Ohio enumeration workshop that I did a few years ago. We settled that they would adopt settling or filtering samples in the Utermalt chamber if concentration is needed. How do we know if we needed to concentrate? It's that magic number 10. Counting 400 natural units for 10% precision because they're usually dealing with colonies of Microcystis or Anabina. Counting cells in 10 natural units for cells per mil, with large colonies can have a portion of, and if they have large colonies, sorry, if they have large colonies, then a portion of the colony is counted and the colony size is measured, because at the same time you will have an ocular grid that actually gives you the size, plus you have the calibration on the grids as well. So if you have to count a colony, a portion of the colony. Now, how much the portion of the colony? Is it 10% or 25%? I don't think we've settled on that, because I don't know. Again, being consistent, I would say you need to go counting 10 or 20% of the colony, and then you measure the size of that colony, and just estimate the total number of cells in that colony. All right. Well, maybe you have a program that says I need something else besides numbers. What if I need biomass? And that's really where it gets tough, but it's doable, because essentially what you're doing then is taking those cell counts and converting that to a biovolume of so many, cubic nanometers per mil or something like that. Why do we need that? Well, I don't know. You have to decide whether you need it. If you think the reasons you might need it is that you could have a fairly small organism that's the toxin producer, and if you just did cell counts, you might misrepresent the totals in terms of total cell counts, what was there contributing to the toxicity. So sometimes biovolume is important to have. And as you saw in that RFP from the city of Salem, they wanted biovolumes. Well, in terms of doing biomasses, you can do biovolumes or you can do biomass. And one nice thing about algae is that everything I've read is that algae have about a specific gravity of 1. And so that helps you in your biomass determinations. And then the other thing too, if you have biomass or biovolume, then you can estimate carbon. And sometimes, especially for nutrient studies and whatever it is, it's important to have, what's the carbon contribution in this system from the algae. Now, these kinds of things probably are not going to play into what you're doing for toxins and risk, but biovolume might. And having biovolumes, you can convert to biomass because of the specific gravity of 1. So biovolume estimates allow you to determine the importance of individual taxa based on their size and abundance. And that sometimes is important. Then you can calculate the volume of the living protoplasm based on common geometric figures. So how are we going to determine the volume of a cell, right? Well, think of it this way. Geometric shapes. We can determine volumes of basic geometric shapes with fairly simple formulas. So what we're going to assign is a geometric shape to the cells. Whether they're circular, spherical, then we have a volume. Whether they're cylindrical, whether they're a cube. And of course, when you think, most of the ones we're dealing with, Microcystis is cylindrical, is a circular sphere. The Anabinas and the Aphanizomenons are cylinders. So we adopt those geometric shapes. And of course, you're always excluding sheaths and spines and things like that, which we aren't dealing with cyanobacteria anyway, except for the sheaths. So we're just looking. And the other thing is, remember, we're going to be using a formula, so all we have to do is have cell counts, and then we plug that into a formula. We're not determining individual biovolume. Biovolume calculation references are in a number of locations, and there are some tables that all you do, if you have 100,000 Microcystis cells, you go to this table, and it will tell you what that biovolume is. So again, all you have to do is have the numbers, and you get the biovolumes from tables. Those tables, there's one on the NAQUA, which is the USGS NAQUA National something Quality Assessment, on the ecology of freshwater algae data files. They will give you the tables for biovolumes. We discussed biovolumes in the toxic cyanobacteria handbook that I mentioned, but the other ones that I like in particular, there is a biomass standing crop in the standard methods examination of water and wastewater. There are tables in there for biovolumes. In Europe, the one that I've compared this one with this one, which is one that's done in Scandinavia, 2006, biovolumes and size classes for phytoplankton from the Baltic Sea. And to show you as an example, using Aphanizomenon flos-aquae, what you can find. So, biovolumes are obtained from cell counts by determining the average cell volume for each species or unit, multiplying that value by the cell number. This results in the total volume for each species. Since plankton have a specific weight of about 1, this biovolume corresponds to the biomass. The formula used for cell volume based on the basic geometric shape of the cell. So Aphanizomenon flos-aquae would be the cylinder, and that equation would be the equation for a cylinder. And you don't even have to do that because you go to the table, okay? So those two references were used to give the biovolume conversions, the OLINA and the from the NAQUA program in this case. Using the OLINA method, we had 196.3 cubic micrometers per cell, and the USGS NAQUA gave a value of 205, pretty close. So if we were counting Aphanizomenon, we would use 200 micrometers per cell for the volume, which also corresponds to the biomass if you wanted biomass. So not impossible, tedious, but doable if biovolumes are important, and they very well might be. Now, at the levels you're starting out, maybe not. You'd probably be lucky to get funding just to do enumerations and toxins, but it is doable. Just to show you again what this is all about, here's the sphere, which might be microcystis. Here's the cylinder, which could be anabenas. Here's a rectangular box that was closed in a sheath. This is glucapsic, so we could use the formula for a box, a rectangular box in this case. Or the screw-shaped ones like Cylindrospermopsis or some of the anabenas, likewise you could use the formula for a screw shape, spiral shape. There are geometric formulas for that one as well. So we're counting the cells in here, using that formula, and getting biovolume. Makes sense? Yeah, it's easier than, but it's not impossible. Harder than it might look, I guess. What about optics, briefly? I guess the main thing here is that bright field doesn't do a very good job. If you're counting zooplankton and things like that have lots of shape and bit of color to them, but when you're looking at cyanobacteria, you need phase because they're prokaryotic. They don't have a lot of shape. You need the contrast that phase gives you. So bright field really doesn't have good enhancement, produces little detail. Phase is about as basic as you can use. And essentially what you're doing is adjusting an annulus on the scope to sort of scatter the light a bit through different angles, so you get a better sense of the shape and the detail in the cell. And I think I was told that all these scopes have, they're usually now on all of the standard scopes that are in, even a teaching lab. But if you have the money and a little more information, something called differential interference contrast, or Nomarsky DIC, actually uses prisms as beam splitters, and it literally gives you a 3D effect. And that would give you the best. But, you know, to adapt a scope for Nemarsky is several $1,000s in itself. But, you know, you can use it a lot. But phase usually gets you what you need to know. And then sometimes efflorescent attachments to scopes work too, because a lot of algae have natural fluorescence, and cyanobacteria are very good at this. In fact, if you had a field sample and it was full of different kinds of algae and they were all round, epifluorescence would actually light up the cyanobacteria, and you could see which ones were cyanobacteria. But again, epifluorescence adds a few $1,000s to the cost of the scope as well. But about the minimum you need is phase. Yeah. Do you have a question?
So could you like get like a UV flashlight or?
Oh, dark light, black light or something?
That would just like shine it on while you're watching?
I don't think so, because you need to really get it underneath and coming up through. Yeah. I'm not sure. I've never tried that. So, yeah, $20 black light. The other thing about the epifluorescence is you need certain wavelength filters, and they're not necessarily the black light filter. So no, you're probably going to have to go to that. So here, for example, blue excitation causes chlorophyll to fluoresce back red. So you can actually distinguish algae from other round things in the sample. Green excitation causes phycoerythrins to fluoresce back bright orange-red, and phycocyanin to fluoresce back orange-yellow. So that's how you would distinguish, because only the cyanobacteria have those phycobilisomes and the phycocyanin, phycoerythrin pigmentation. So, a couple of educational sites for troubleshooting microscopes. And then how do we use this information? Cell counts, biovolumes, etc. are obviously going to be used to set some kind of guideline and either determine alerts. It may also involve toxin analysis, but not necessarily. This is just a sample then of the flow chart that we developed for the World Health Organization book, this one. So it's in that book. So you're examining, you're microscopically examining cyanobacteria detected at low levels, one colony, which would be a natural unit, or greater than 5 filaments, which again would be natural units. Yes or no, and then of course, all it took in this case was 1 colony or 5 filaments to get us to this level where we start monitoring more regularly, and then you start asking more questions like how many cells per mil, or how much chlorophyll, or in this case, how much biovolume, to push you to the next level, and so on and so forth. And at some point then you have to decide to do cyanotoxin analysis. So these are the kind of flow charts, this one, and most states have them. This is what Oregon put together out of their program, you know, kind of a more simplified thing, but anywhere. You know, visible scums with documentation and testing, photos, issue advisory if no. Then option 2, toxigenic species combined with cell counts. Their cell counts were greater than or equal to 100,000 cells per mil. If that exceeded, then it went to the advisory. Keeping in mind that everything's advisory, there's no regulation in this. Then toxigenic species, individual species. This would be combined cell counts of everything that's in there. And individual species, for example, on the more possible, probable toxigenic ones like microcystis or plankton, then you only need 40,000 cells per mil to issue the advisory. And then the other option, toxin testing. So that's, again, how you generate these flow charts for your decision-making process. And putting those all together, this is this is out of the WHO guideline and Oregon state of Oregon, I should say. Quantity of cells, type, frequency consideration, public info, and outreach based on different cell counts. So again, you could approach it that way too. And in the case of the USEPA, again in June 2015, this is their new advisory. So health advisory, EPA recommends health advisory levels at or below now 0.3 micrograms per liter for microcystins and 0.7 micrograms per liter for cylindrospermopsin in drinking water. That would again be the finished drinking water. For children preschool age and younger, the main difference that the EPA has done that we didn't in the World Health Organization was distinguish between children and adult exposure. For the World Health Organization, we just went with everybody because in developing countries, it doesn't really matter. But here in the U.S., they wanted to distinguish. Of course, if you think about it, what water utility is going to adopt advisories just for adults? So you're automatically forced to drop down to the 0.3, which effectively is making the guideline values now a third lower than what they were before. Okay, recommended levels though for adults were raised to 1.6 and 3 for cylindrospermopsin. So this is what's now in the US EPA documents, and I assume that most states will at least consider that if they are developing their own. California is, Beverly, is it, what is it? I guess it's 1. Well, California's 1, yeah. And then the US EPA, federal agencies, for all of their risk management stuff, whether it's arsenic or microcystins, they like to use the stoplight approach to guidance. So low-level microcystins, 0.3 or below is a green light. 0.3 to 1.6 is yellow, and red is above 1.6. So that's how they're, that's again in these new documents that you can download. Okay, I was told again not to talk about water treatment, but then at the same time sort of cover it. So I threw in a couple of slides. Excuse me, water. Treatment methods for cyanotoxins. And this comes mostly out of Europe and Australia, and to a certain extent South America and the U.S. But this is the AWWA white paper that condensed all of this. And in general, their statement is that powdered activated carbon, dose required varies with the water quality, whether you have a lot of color in your water or pH and things like that. But for toxins, the extracellular toxins, microcystins, except microcystin LA seems to be okay. In other words, PAC for microcystins is okay. Caveat being that wood-based chemically activated carbon is the most effective. 60-minute contact time. I remember when they say wood-based, what they're referring to, a lot of activated PAC carbons use coconut shells is another big source of carbon used in water treatment plants. Those aren't as effective. Cylindrospermopsin and saxitoxins, again, PAC works, but for LA you have to have really high doses, maybe more impractical that you wouldn't typically use. For cylindrospermopsin, wood-based chemically activated carbon is the most effective. Again, a 60-minute contact time. Saxitoxins, the recommendation is a microporous carbon, coconut or coal-based, steam-activated wood, 60-minute contact time. Granular activated carbon, dissolved toxins. GAC absorption displays a limited lifetime for all toxins, varying between 2 months to a year, depending on the type and the water quality. So in reality, powdered PAC is better than GAC, even though, of course, GAC is cheaper. Biological filtration. What does biological filtration mean? Biofilms. The Japanese kind of pioneered this. They actually developed these biofilms, these big plates that are placed in the water treatment stream. The water flows over those, and organisms on that actually inactivate the toxins. And they get these natural populations that are composed of different kinds of protozoa and other ciliates that seem to be able to handle the microsystems. And they like it, but again, it's on a smaller water treatment systems that seems to be best. Membrane processes, these are the more advanced ones, maybe reverse osmosis, which of course would be very expensive, but might be necessary. And advanced oxidation. All of the advanced oxidations, whether they're ozone or different kinds of chlorine enhanced or permanganates or things like that, work, but you have to adapt and adopt them to particular toxins and your water quality issues. So, in summary, treatment effectiveness, chlorines, show you across there, most effective with microcystin, not so effective with the alkaloid toxins. Chloramines, not much effect all the way across. Chlorine dioxide, not very good. Potassium permanganates, effective on antitoxin. In general, it's reasonably effective. Ozone, effective except for saxitoxins. And UV advanced oxidation, not effective on microcystin. That's primarily that cyclic peptide doesn't break down under UV, but effective against these two toxins. So that's kind of the summary that I have for water, out of the water treatment. The references for that are the following. American Water Works Association, this document, Optimizing Conventional Treatment. This is Australia. And then there are chapters 9 through 14 in the big USEPA book, too. Okay? This is the lake in Oregon that I've been helping watershed councils with. And actually, we have samples back there that I want to use in the ID part to show you how to use the dichotomous key. So that's why that slide is there.